Hello everyone. In the last class, we have discussed about the monocistronic and polycistronic mRNAs, as well as we have seen about the simple concept that is gene duplication, how it occurs during the meiotic crossing over. So in today's class, we're gonna discuss about the simple and multiple transcription units. We know well about the transcription. So for the synthesis of a polypeptide chain, or for the synthesis of any small RNAs or tRNA or rRNA, we need a sequence that is transcribed from the DNA. Okay, so if you see the bacterial transcription, so most of the bacterial transcriptions are the like operon concepts, like we have seen lac operon, right? So in the lac operon, a single mRNA contain three type of cistrons which can encode for the three types of enzymes. Okay, the cluster of genes that forms a bacterial operon constitutes a single transcription unit. So if you see bacterial operon, example lac operon, it contains a single transcription unit. That means during transcription, it will bind to the specific promoter and it will start the transcription from the initiation site, and it will continue the transcription up to the termination site. So in between this initiation and transcription sites, there are three genes which encode for the three enzymes in the lac operon. Okay, so that means a single primary transcript can able to encode three enzymes. But one thing we have to understand, that the bacterial transcription and translation is a simultaneous process, because the transcription and translation will takes place simultaneously, one after other, immediately. But one thing is that when come to eukaryotic mRNAs contain the introns. So these introns are the non-coding sequences which are not useful for the translation. So that's why eukaryotic mRNAs, first after transcription we will get an mRNA which is called a primary transcript. That primary transcript undergo post-transcriptional modifications such as capping, splicing and polyadenylation, and finally it will become the mature mRNA. Okay, so eukaryotic transcription units, however, are classified into two types depending on the fate of the primary transcript. Usually eukaryotic transcription units are two types. It's depend upon the primary transcript. One is the simple transcription unit and another one is the complex transcription unit. So for example, if you see the simple transcription unit, example beta globin. So the primary transcript which undergo the post-transcriptional modifications, we will get a single type of mRNA that encodes only single protein. But 90% of human transcription units are the complex process, are the complex units. So if you see, the primary RNA transcript can be processed in the more than one way, because if you see, there are different types of exons which we can encounter in the eukaryotic complex transcription units. So using alternative splicing mechanisms, so the machinery will join the different exons and will produce the different type of proteins using the same primary transcript. Okay, so when we see this alternative mRNAs which are produced from the alternative splicing, so these all are the monocistronic mRNAs. These alternative mRNAs are the monocistronic mRNAs that encode one type of a protein or one type of polypeptide chain only. But every alternative mRNA which is produced from the primary transcript must contain the AUG, which is the first sequence can be decoded by the translation machinery. So it will add the methionine as the first amino acid. Okay, when come to the complex transcription units, so from complex transcription unit we will get the multiple types of mRNAs. Okay, so it usually takes place in the three ways that we will describe in the picture. So examples for this all three types of alternative RNA processings is the sexual differentiation genes in the Drosophila. So the Drosophila genes which regulate the sexual differentiation can undergo this alternative type of RNA processing. Commonly, one mRNA is produced from a complex transcription unit in some cell types and a different mRNA is made in other cell types. See, one cell requires some kind of amino acid sequence and within the same protein, and another cells require another type of amino acid sequence. So within the same protein. So using alternative splicing mechanism or alternative mRNA's production, so the cells can get the or can achieve the required amino acid sequences. Okay, so for example, if we see the fibronectin transcript, so the fibronectin transcript, that is nothing but the primary mRNA of a fibronectin protein in fibroblast cells as well as hepatocytes. If we see the alternative splicing mechanism, so it will determine whether the cell surface binding domain should contain or not. So for example, if you see this fibronectin protein sometimes may have the cell surface binding domain, that is binding part, and sometimes it will never have the binding part because the exon part which encode for the future amino acid sequence will be removed by the alternative splicing mechanism. So there is absence of particular binding domain. So the relationship between a mutation and a gene is not always straightforward when it comes to the complex transcription unit. For example, if a gene is mutated, maybe at the regulatory sequence or at the coding sequence. So we can say if coding sequence is changed means amino acid sequence will change. If the regulatory sequence is mutated or changed, automatically what will happen? The regulatory region unable to recognize it by the RNA polymerase and there is no transcription, or else sometimes the transcription may occur in a different way. Okay, but when come to the complex transcription units, if a mutation occurred in the control region or if a mutation occurred in the exons, so automatically what will happen? All the alternative spliced mRNAs will change. So it's not only about a single mRNA, because in the complex transcription units we will get many type of alternative mRNAs. So every mRNA will affect by these mutations. Okay, so genetic complementation tests are commonly used to determine if two mutations are in the same or different genes. So with the help of genetic complementation we can determine a mutation which is that is two mutations that are occurred in the same gene or in the different genes. So that we have already seen in the genetics in an academy platform. So these proteins, so for example we have discussed that there are three types of alternative mRNA processings will occur. So each will produce different type of polypeptide chains, but these all formed from the single primary transcript. These all formed from the single primary transcript. That's why we will call it as isoforms. Okay, these all are called as isoforms. So you can see here complex transcription units. If you see first type, so there is a cap site here. The A and B are the regulatory sites, and the cap site, you know, eukaryotes having the capping site and the polyadenylation site. So if you see the this primary transcript, it having the 4 exons. Okay, it having the 4 exons. If you see the first mRNA which is produced from this primary transcript contain the all the exons. From cap site to polyadenylyl site, it achieve it to the matured mRNA. But sometimes see here in the second mRNA there is a lack of exon 3. Maybe that amino acid sequence is not required. If you want to add amino acids, we need a lot of energy. So it is reducing the energy by removing the unnecessary nucleotide sequence that is not useful for that particular function of this mRNA. Okay, I mean this mRNA will convert will translate into polypeptide chain. Maybe that polypeptide chain does not need that kind of sequence for its functions, so it can reduce the energy utilization. So if you see the gene 2, it having a single cap site but two polyadenylyl sites. So two polyadenylyl sites means you can see directly. So these two exons can be transcribed or these two exons can be transcribed. Okay, so use of polyadenylyl sites more means we can terminate the translation in between. Okay, so where the polyadenylyl site is there. So we have to remember for a primary transcript, if it is undergoing post-transcriptional modification, it should contain the cap site as well as polyadenylyl site that is required for the future translation. Okay, and if you see gene 3, so the gene 3 contain two cap sites. So the alternative splicing may initiate from here or else from here. If it is initiated from here, then it will leave this exon, that is 1b, and through alternative splicing it is achieving the exon 2 and 3. But if the alternative splicing started from exon 1b, then exon 1b, exon 2, exon 3 will achieve or obtain in the matured mRNA. Okay, so in complex transcription units, it is just an alternative splicing mechanism. With the help of alternative splicing mechanism, it will attain or it will achieve the different type of amino acid sequences. So reducing the unnecessary sequence also can reduce the energy utilization. So if you see the simple transcription unit, there is a cap site and there is a poly A site, directly the mRNA is transcribing. Okay, so the black color lines which are in these genes are the intron sequences that are usually removed during the splicing mechanism. So this is about the simple and complex transcription units. So in the next class we will see the gene family. So that is a very important concept.